Thank you everyone for standing by and welcome to our webinar entitled Project Cyborg, Assessing Limiting Factors for Diverse Cyanobacterial Blooms and Their Associated Toxins. This webinar series called Freshwater Science highlights Ohio Sea Grant partnering scientists every month. Every quarter is a different focus, from human health and fish farming to harmful algal blooms and human decision-making, bringing applied research to the public on issues that affect our Great Lakes communities. I am Jill Gentis-Banicki from Ohio Sea Grant and Stone Laboratory, and joining me today are Dr. George Boullerjean from Bowling Green State University, Bob Sterner from University of Minnesota Duluth, and Todd Miller from University of Wisconsin-Milwaukee. Dr. Boullerjean is an emeritus professor at Bowling Green State University in Ohio and part of the Great Lakes Center for Freshwaters and Human Health, where his team currently examines the composition and dynamics of cyanobacterial and microbial communities in freshwater environments, focusing primarily on nitrogen and phosphorus cycles in the Great Lakes. Dr. Bob Sterner is at the University of Minnesota Duluth, where he is the director of the Large Lakes Observatory, the only institute in the world dedicated to the scientific study of all the large lakes on Earth. His recent work there includes lead authorship of a paper on the grand challenges for research in the Great Lakes, the first paper examining cyanobacterial blooms in Lake Superior. Dr. Todd Miller is within the College of Public Health at the University of Wisconsin-Milwaukee. His lab is particularly interested in understanding how microbial communities affect the concentration, fate, and toxicity of harmful chemicals in the aquatic environment, with the ultimate goal to formulate models of toxin production, release, and degradation in aquatic systems. We're really delighted to have everyone here, these three researchers, and a collaborative project with Wisconsin, Minnesota, and Ohio Sea Grant funding these research projects. Before we do anything, I'd like to just mention a few things about the webinars themselves. During this presentation, all participants will be in a listen-only mode. Afterwards, around 12:45, I'll conduct a question and answer session. If you would like to ask a question during the presentation, please feel free to pull up that chat feature at the bottom of your screen during their talk, and I'll collect and pose your questions out to each researcher at the end of their presentation. As a reminder, this webinar has auto captioning and is being recorded to be posted onto our webinar or website for later viewing. Also, we will post a webinar survey in the chat feature toward the end of the hour. Please take a few minutes after the webinar to fill out that survey. It always helps us continue to bring you great webinars. So without any further delay, I'd like to introduce Dr. Boullerjean, Dr. Sterner, and Dr. Miller, who will be presenting Project Cyborg, Assessing Limiting Factors for Diverse Cyanobacterial Blooms and Their Associated Toxins.
Great. Thanks so much, Jill. And I also really appreciate the fact that Sea Grant got together, the three different Sea Grants, to fund this program. I think we've all learned a lot, and we're very pleased to provide our progress report on what we've been doing. So, but it has not been I need to advance the slides. This is not just a lonely struggle of us three. The heavy lifting has been done by students and technicians at all three institutions, and we're grateful for their help. And the data we'll be presenting here is really due to their terrific efforts. So what we'll be doing today is providing some data that we've gotten to date. I think there's some nice emerging stories to tell here. First of all, I'm just going to provide an introduction, just talking about the objectives of the project and the overall experimental plan. I'm going to turn the mic over to Bob, who will talk about some bioassay experiments that occur over a short duration. Then back to me, I'll talk about some of the longer bioassay experiments. And then Todd will talk about the toxin analyses from our experiments. And then we can open up the discussion to questions from you all. So obviously the seminal event, at least for the Erie folks, was the 2014 Toledo water crisis. Everybody is aware of that. It is an issue which occurs routinely, almost yearly, and yielding a year-over-year cost, which is significant. This isn't really the topic we'll be talking about, but certainly there enfolded into this is an environmental justice issue, and the impact is primarily in poorer communities. However, Erie isn't the only place where blooms happen. Certainly, there are well-documented sites in Lake Michigan and Lake Huron, but a real shock to us, I started working in Lake Superior with Bob about 20 years ago, and it was unthinkable that blooms would happen there, but now they are happening. And we're seeing Dolichospermum blooms in nearshore environments. And so this is something that really required 3 Sea Grant projects coming together to in one big project to really look at what are the drivers of the different bloom types. When we talk about the relevant bloom forming cyanobacteria, I'm sure many of you are familiar with these organisms. Microcystis being really sort of the main guy that people talk about, at least in the lower Great Lakes. But certainly we're seeing Dolichospermum, the Planktothrix in embayments in Lake Erie, and then Aphanizomenon. And it's come to the attention of the community that Aphanizomenon and Dolichospermum are different morphotypes of the same clade. So I'll often be talking about the ADA clade when I talk about the longer bioassays. The toxins that are produced, the threats are liver toxins and neurotoxins. And they're really, you can view cyanobacteria as factories of secondary metabolites, a lot of which remain uncharacterized, and what their bioactivity may be still requires further examination. And so there's a lot of interest in what these compounds are and who's making them. And so different bloom types occur throughout the Great Lakes. If you're looking at Lake Superior, we're talking primarily Dolichospermum, Microcystis, and Green Bay. And then Lake Erie has sort of a smorgasbord of things that can bloom: Microcystis, Dolichospermum, and Planktothrix, most notably. And so driving our project is the hypothesis that nutrient stoichiometry between nitrogen and phosphorus and temperature interact to steer bloom toxicity, as well as the identity of dominant and interacting species. Basically, how does nutrient stoichiometry structure bloom types, and what are the different bloom types, and what are the toxins that these blooms produce? The lakes have different geochemical profiles as you go from the upper lakes down to the lower lakes. Lake Erie certainly has a much lower N to P ratio compared to Superior. Michigan is sort of in between. And if you look at our initial chemistries from the assays that we've done, this sort of parallels that nicely, in that Lake Superior has a TN to TP that's much higher than our samples from Sandusky Bay, Lake Erie, and with Lake Michigan being sort of in between, with Green Bay. This is Green Bay, Mowickwi Bay, and Sandusky Bays of the individual lakes. And so there has been a published scheme for bloom formation proposed by Chris Gobler back in 2016 in Harmful Algae, where really you could look at gradients of phosphorus and nitrogen as structuring communities. And we will see in our initial work that this will sort of hold true when we're talking about manipulating nitrogen and phosphorus concentrations with our Erie samples. And so a major driver of the different bloom types are the ratios of phosphorus and perhaps different species of nitrogen. So when we consider how these blooms form, we consider the environmental gradients that we just talked about with respect to nutrients. Also, we have to consider climate warming, and that affects not only warmer water, but more rainfall, which may drive more nutrients from the landscape into the lake. And how we're addressing this is with bioassays of different duration. And samples from these bioassays can subsequently be analyzed for toxins, for phylogenetics and genomics, and also just total biomass. And so we will be providing some of these data to try to tell the story that we have today. And so our experimental approach to the use of bioassays, this is our hypothesis, as I stated earlier, but we'll be adding, taking water samples to be amended with the different nutrients, different nitrogen species, along with phosphorus. And there are two types of experiments. The first one is to answer the question, what is or are the limiting nutrients? That would be short bioassays in which different N species are provided either alone or together, and also in concert with phosphorus and then phosphorus alone. These last for 48 hours. Then we have long bioassays, which are in large jugs occurring over a 28-day period, basically asking who wins, what cyanobacteria dominate and bloom. And these will run at ambient temperature and elevated temperature. And then the bioassays are sampled by a number of parameters to try to figure out not only what who's there, but what are they making. The phylogenetic assessment through DNA sequences just 16S thus far. The omics work is currently ongoing. A big thing is that all protocols in all three labs are done the same way. So perhaps this is something that other labs could be doing elsewhere in the world, where we could do comparisons of different lakes, because we devised and documented a very precise protocol for how these experiments should be done to allow lab by lab comparison. Okay, so for the short-term bioassays, I think Bob is going to go in a little more detail than this but we add individual nitrogen species and triplicate to bottles, or an all-N treatment, either with phosphorus or phosphorus alone. And of course, there's a no nutrient amendment controls as well. The long-term bioassays, this results in a lot of bottles, 30 bottles at ambient temperature, and then sort of an abridged set of treatments, adding up to 12 bottles in the short-term bioassay. The long-term bioassay are 4-liter jugs in duplicate, in which we're adding either phosphorus, all the nitrogen species, and then phosphorus and all the nitrogen species in concert. To keep the bloom forming and developing in the bottles, every 3 days, 1/10 of the volume in the long bioassay is replaced with filtered lake water, appropriately amended for the given bottle we're talking that's being used. And then so the samples are then samples are then analyzed as listed here. Samples are taken from Lake Erie in Sandusky Bay, Lake Superior in Mawikwi Bay, and then a buoy location in Green Bay, monitored by Todd Miller. Okay, so Bob, I will pass the mic over to you. I will have slide control, so feel free to just tell me to advance slides when needed.
Great, thanks, George. Yeah, so George already showed this. Go ahead and click it. Again, the question with the short bioassays is sort of what's limiting. And of course, there's a whole bunch of literature about what you can and can't do with these short bottle bioassay types of studies. I'm not going to get too bogged down into that, but the general framework is that regardless of whatever complexities there may be, if you take a community from the lake and over a short window of time, 2 days in Green Bay and Lake Erie, 4 days in Lake Superior, over that short period of time, if you spike these bottles with different substances, the algae will tell you something about what they're potentially short of, and respond to whatever spikes they will respond to. You saw this design already. It's actually sort of 2 nested experiments. I'm going to be talking a little bit about both. Go ahead and advance it, George.
Yep.
I don't want to get too bogged down in this, but it's important in the back of your mind when we're looking at, do the algae respond and how much do they respond? That's related to two different things. Essentially, how fast they're growing under ambient concentrations, which is, I guess my cursor doesn't work, George. That's a failure of this.
Oh, yeah,
Yeah, how fast they're growing under ambient conditions, that left vertical line.
Yeah.
And then how fast they're growing when nutrients are added. And so that difference can be small or large depending on how limiting nutrients are, but also how fast algae can grow when they're given a lot of nutrients. So that's just something to keep in mind. I also want to mention here, I'll show you a plot or two of something we call response ratios, which is a calculation that you make from comparing the biomass in a treatment divided by the biomass in a control, and then you take the log of that and divide by time. Okay, so the data. Big picture, the lakes act like many lakes do, where you get some response to either nutrient alone, and often, although not always, the largest response to both nutrients put together. Sandusky Bay on the left, you can see 0 or 1 P. Whether there's nitrogen or not is in color, so no nitrogen's red, with nitrogen's in blue, and ambient and elevated. And your eye will tell you from that left graph under Sandusky Bay that they generally did best with both N and P and at elevated temperature. Green Bay was quite a bit different in that they did do better with both N and P, but they did not like the elevated temperature, and we're still working to try to understand what how to interpret what's going on in Green Bay, where the relationship of the algae to nutrients changed really dramatically by us just warming the bottles up a few degrees. And then Mowickwi Bay and Lake Superior they're responding mostly to phosphorus, but also to the elevated temperature. Next slide, please. So this is where we talk about these response ratios, and without getting into the math, the horizontal axis is basically a measure of how limiting was phosphorus, and the vertical axis is a measure of how limiting was nitrogen, and the black diagonal line is the 1-to-1 line. So points above the diagonal are where the algae responded more to nitrogen, and therefore you could say we're more limited by nitrogen. And below the diagonal is algae were responding more to P, and therefore you could say we're more limited by P. And the broad picture here was what we expected, which was Lake Superior algae were more limited by P than by N, although some modest increase in biomass from adding N. The pattern in Green Bay and Lake Erie to me is super fascinating because the dots are all over the place, generally above the diagonal, but the seasonal variability here is a really important part of this story. And this analysis is giving us a lot more information than we had before about the seasonality of these different forms of nutrient limitation. So we talk about the blooms being steered. And so at each point here, that's a separate bioassay run. And this is telling us a little bit about how the environment is steering left or right, or nitrogen or phosphorus, depending on what's going on. Next slide, please, George. I hinted around a little bit about seasonality. And so the left graphs are plotting seasonality in the response ratios for phosphorus on the top and the response ratios for nitrogen on the bottom. And the most apparent pattern here is for both Green Bay and for Sandusky Bay, the blue and the kind of brownish dots, phosphorus limitation was strong and especially associated with early in the season. By mid-season, that's day of year on the x-axis. By mid-season, when typically these environments are really cooking with blue-green blooms, the phosphorus limitation goes away, but we still see nitrogen limitation. But the nitrogen limitation has a more complex seasonal pattern, and it's driven more by just the concentration of nitrate in the water. And that's what the rightmost graph on this slide shows. And so the nitrate isn't smoothly changing through the season. It's going up and down. There are some seasonal patterns, but it's not a smooth seasonal change in nitrate. It's jumping up and down in these environments. And whether the algae are limited by N or not mostly seems to depend on what the sort of present moment concentration of nitrate is in the water. Next slide. Temperature is also an interesting and sort of complicated story. So these are these response ratios for temperature, where we're dividing the biomass observed under elevated temperature by the biomass observed under ambient temperature, and by log and day. And so it's giving us a quantitative indication of, for values above 0, how much faster did the algae grow when we warmed them up a few degrees, like is projected in, let's say, the coming decades. And the solid black bars are the most important ones here. Those are the ones that are the temperature response in the absence of any added nutrients. So just warming. And you see, in all three environments, the black bars are not always, but usually greater than 0. So a little bit of warming, even just 3 degrees, gives us measurably higher biomass and therefore faster growth. Where at Green Bay, for some reason that we don't quite understand yet, those black bars are really far to the right. So warming in the absence of nutrients really jacked up the Green Bay algae. At the same time, the gray bars are response ratios to temperature with nutrients added. And there the Green Bay story is really complicated, in that adding nutrients often shut off or in fact reversed the effect of temperature. So there's some unexpected interaction of temperature and nutrients going on just in Green Bay that we're still working to try to understand. The mean increase for 3 degrees, like we project in a few decades from now, the mean increase in biomass corresponds to about an extra doubling of the algae in a 9-day window. So in that future world, everything else being equal, which of course it never will be, but these experiments work variable by variable, all else being equal, in a future warmer world, doubling times for algae will be on the order of, the question in the chat, 3 degrees Celsius. And so this will increase doubling with about a 9-day time frame. Okay, next slide. I might be close to the end. That's it for me.
That's it. That's
See
it
you
for
at the
you,
end.
Bob. Okay, so back to me regarding the long bioassays, and basically these are the experiments which ask who wins and what cyanobacteria respond in bloom in response to these nutrients over the long term, and what nutrients structure the communities that result. And so just looking, this is principal coordinates analysis, basically looking at the dissimilarity of the communities from by 16S data, looking at all bottles, all lakes, and all time points from the long bioassays. And this just shows that the 3 communities are different. This includes cyanobacteria, chloroplasts, and heterotrophic bacteria, which are present. So the 3 communities are different. So let's look and see within these communities how the cyanobacteria communities may change. And so, but before we do that, we can talk about chlorophyll. We can focus on superior. And based on what Bob says, it probably comes as not a huge surprise that nitrogen, phosphorus has a very large effect. Nitrogen has as in these experiments, basically no effect. And together, nitrogen and phosphorus, it's not, in these experiments, not statistically significantly different. So phosphorus is obviously the nutrient to which the community is responding with respect to total biomass. If you look at the 16S data, we sampled at 14 days and 28 days, and the elevated temperature are the right-hand sides of each of these arrays. Okay, so these and this is bottle number down here. What we see, knowing aware that the biomass is changing drastically or much greater upon phosphorus amendment. What tends to happen in these bottles, we have Dolichospermum present, and that Dolichospermum, it's binned here as a Phanosomenon, but under the microscope, it's Dolichospermum. Dolichospermum does, the ADA clade, including Dolichospermum, takes over. We know that the biomass is driven by phosphorus, but so phosphorus appears to structure the community, at least just at looking at biomass in this graph here. Also, it looks like at higher temperatures, we're seeing a more diverse community. So perhaps Dolichospermum may prefer lower temperature, but certainly more work needs to be done to determine that. Now, we see Dolichospermum, this is work by Rianne Loisel. We see higher relative abundance with phosphorus amendments at both elevated and ambient temperature, and then starting out with an initial here, and we're seeing an increase at both time points, just with phosphorus amendments and ambient with all nutrients added. Okay, both ambient and elevated. Now, if we look at the principal coordinates analysis of just the cyanobacterial 16S data, it's a fairly clear-cut analysis. We can look at the different treatments, which are color-coded over here, and the time points that are over here. We can break this down a little more simply in that all the time points from 14 days populate the bottom half of the figure. All the 28-day samples populate the upper half. This is our initial sample right here. And then if you look at the nutrient amendments, all the no addition or nitrogen amendments populate the left side, and then anything containing phosphorus populates the right side of the figure, which basically the conclusion I draw from this is that phosphorus is structuring the cyanobacterial community, which is developing during the course of the experiment. Dolichospermum wins, and if we want to bring it down to a management perspective, management of phosphorus is appropriate when we're dealing with these blooms that may occur in Superior. With Sandusky, it's a little more complicated and unexpected. A lot of this is very seasonal with respect to Lake Erie. We're only doing a limited number of these long bioassays. So we took a sample at sort of a midsummer bloom period when N to P begins to drop. And there are a number of mechanisms by which we see nitrogen losses. There's denitrification, there's biomass, there's assimilation of nitrogen from by the biomass. But here the T N to PP was about 24 at this date. If we look at the ambient temperature, amendment with nitrogen in the long bioassay yields a positive response. However, at elevated temperature, phosphorus has an effect. We see this at both 14 days and 28 days. And so it's kind of hard to explain this, but we also have to remember that cyanobacteria aren't the only things in the bottle. There are other algae and heterotrophic bacteria which are also likely competing for nutrients, and perhaps the competition for hand waving, perhaps the competition for nutrients for phosphorus or for phosphorus is greater at higher temperature. Now, if we look at the long bioassay 16S data, if this is the initial, and then if you just looking at some of these columns, these are duplicate bottles, and it looks like there's some decent, just by eye, pairwise reproducibility between the bottles. So the nitrogen amendments are here, phosphorus amendments are here. One thing you can tell at a glance, not in addition to some decent pairwise reproducibility, is that there's higher relative abundance of N fixers in most phosphorus amendments, or you could call it suppression of N fixers and nitrogen amendments. There's a major community shift at elevated temperature and a mixed community. So it appears that it's a mixed community responding more to nitrogen than to phosphorus amendments. If we look at Rianne's floristics, Dolichospermum is responding to nitrogen, to phosphorus. Microcystis is responding to nitrogen amendments. And so here we're dealing with a nitrogen fixer responding to phosphorus. We're dealing with a non-diisotrophic responding to nitrogen. Looking at the PCOA from this, there's again a reasonably clear-cut differentiation of the samples. This is our initial here, and the early time points populate the bottom half of the figure. The later time points populate the upper part, indicating a bloom is developing during the course of the experiment. And then phosphorus and no amendment is all over here. Nitrogen or phosphorus in concert with nitrogen populate the left side. And so this indicates that nitrogen is more important at this point in time structuring the cyanobacterial community. A long bioassay taken earlier in the season when the N/P ratio was different would likely yield a different result, because we're seeing a great deal of seasonality with respect to the short bioassays. But in the Erie mixed community, just looking at this experiment in detail, Gobler's model seems to work. Phosphorus amendments seem to favor Dolichospermum. Nitrogen amendments tended to favor Microcystis. And so that was an appealing result that we got. So the summary of the long bioassays: superior P structures the cyano community by 16S, N structures the cyanobacterial community in Sandusky. And this, again, bringing it down to management, looking at the seasonal variability, N and P management is appropriate for Sandusky, and management of P loadings for Superior. Future work will complete the 2023 bioassays. We also see an N effect in the 2023 bioassays. I didn't present it here. I include chloroplast data and all the analyses, toxin analysis, which Todd will be talking about shortly, and then metagenomics on targeted samples. We did see in 2023 a bottle crash. A lot of my lab is looking at mechanisms of bloom decline. And so it would be interesting to see if we are finding any new viruses in a crash bottle, so for example. Anyway, moving on, I think it's now time to have Todd talk.
Thanks, George. So I'll talk about toxin analyses we've done so far from these bioassays. And our first set of analyses targeted peptide toxins produced by cyanobacteria using liquid chromatography, tandem mass spectrometry. We are also looking for neurotoxins. That work is ongoing. I think probably most people know what microcystins are. They're liver toxins that also affect other organs. And there's a wide variety of microcystin structures that are produced in nature. We target 11 congeners and also nodularin, which is a similar molecule that generally occurs in brackish waters, but has been detected in benthic organisms of freshwater environments. And then, as was mentioned by George, cyanobacteria are factories for secondary metabolite production. And so we try to get some of this quantitatively by targeting some other classes of peptide, what I'll call emerging toxins, first looked at for their potential pharmaceutical applications, but now we're seeing that they are in fact toxins. We also, so that's anavanopeptins, cyanopeptolins, and microgynin. So I'll just say right now, we didn't detect any nodularin or microgynin in any of these treatments. We'll be focusing on microcystins, anavanopeptins, and cyanopeptolins. Go to the next slide. So I just wanted to mention, since people might not be familiar with anavanopeptins and cyanopeptolins, they've been known for a long time to inhibit some really essential enzymes in eukaryotic cells, mainly serine proteases like trypsin and chymotrypsin. But anavanopeptin has been shown to inhibit phosphatases as well, and that's the main mode of action of microcystins. So, you know, when you're looking at toxicity of microcystins, if you have presence of anabenopeptins there as well, you could be augmenting that toxicity of microcystins. Both of these have been shown to be toxic to both vertebrates and invertebrates. And in fact, one of my colleagues here at the University of Wisconsin-Milwaukee is finding anabenopeptins to be particularly toxic to human cell lines. And they're produced by a variety of species. We know this now because the genes for the synthesis of these peptides are known and occur in Microcystis, Planktothrix, Dolichospermum, Nostoc, and several others as well. Okay, so here's results from a short bioassay in Green Bay from 2023. And I'll just orient you to these because I have a series of these to show where we have concentration of the toxin. In this case is microcystin in 4 short bioassays on the y-axis, and on the x-axis are the treatments. And the treatments are colored by whether no nutrients were added, N only in blue, P only in red, both in green. Gray is none added. So earlier in the year, and this was kind of goes along with what's been discussed already, on the June 14th, we see more of a response to phosphorus. Perhaps we still have to do some statistical analysis whether or not that's significant or not, but highest concentrations were either with phosphorus or with both sets of nutrients. And then going to the next bioassay in July, and then subsequently on September 8th and 26th, we see highest concentrations in bottles that received various forms of nitrogen. You can go to the next slide. Similar story then with anabenopeptins, where a little bit of seasonality, highest response to nitrogen, but not as great of a difference as if you go into the later year bioassays, where we see greatest responses to nitrogen species and some kind of varied responses to phosphorus or all nutrients. But it seems that addition of nitrogen also increased anabenopeptin production. Go to the next slide. And then we also detected cyanopeptolins in Green Bay. We didn't detect cyanopeptolins from the other lakes bottle assays. And a similar story, although the seasonality for cyanopeptolin wasn't very strong, primarily, again, seeing highest concentration of cyanopeptolin in bottles that received various forms of nitrogen. Go to the next slide. So then here's short bioassay toxin concentrations from Sandusky Bay, looking at microcystins, and there were some varied responses here. So in July 12th, there's really no treatments except possibly all forms of N and all nutrients at ambient or elevated temperature that had increased microcystin production. But if you go to the next bioassay and you look at no nutrients added, the gray box, there was really no effect of adding nutrients on toxin concentrations. And then if you go to the last bioassay on September 27th, N and P alone increased toxin production when both were added. Toxin concentration was quite variable between bottles. Next slide. So here's anabenopeptins from Sandusky Bay. They were quite abundant compared to microcystins, and phosphate plus nitrate addition in the July 12th bioassay caused the greatest increase in anabena concentration. And going to the July 26th, again for anabenopeptin, if you just compare the concentrations to no nutrients added, there didn't seem to be any effect of adding nutrients or elevated temperature for that matter. But on the September 27th, we see the greatest effect of elevated temperature and all nutrients added for anabenopeptin. Go to the next slide. Okay, so Superior is interesting. I didn't really know if we would detect anything, but we did. And anabenopeptin was detected in one short bioassay at fairly low concentrations of 0.03 micrograms per liter. in a bottle receiving ammonium. And it wasn't too surprising to see this, only because we've detected anabanopeptin A in bloom samples that we've gotten from Bob's lab before. I was surprised to see microcystin because we've never seen or measured microcystin in any bloom samples from Milwaukie Bay in Lake Superior, where blooms in Lake Superior have generally happened. And of all the microcystin congeners to detect, it was a very rare one, microcystin LW, but at very, very trace levels in a bottle receiving both nitrate and phosphate, and one bottle incubated at elevated temperature. So what this says to me is that there is genetic potential out there. There are species out there that can make microcystin. Of course, I don't know if it's Dolichospermum or not. Past analyses would suggest it's not, that it might be, Microcystis or some other species that's making it. Go to the next slide. Okay, so here's data from the long bioassay in Green Bay. And we detected, again, microcystin, cyanopeptins, and anabanopeptins in these long bioassays from Green Bay. The production of microcystins and anabanopeptins was highest in treatments receiving nitrogen. So whether it was all species of N or at elevated temperature, and really very little response to additions of phosphorus. There was, in some cases, decline going from day 14 to 24. So it isn't necessarily that the toxin production is sustained. Looking at cyanopeptins, it was highest at elevated temperatures with nitrogen added. Go to the next slide. So here's the long bioassay toxins from Sandusky Bay. We only detected microcystins and anabanopeptins of the peptides that were looked at. And here, again, highest concentrations of microcystin and anabanopeptin occurred in treatments with N, but wasn't necessarily sustained concentrations throughout the entirety of the bioassay to day 28. Next slide. Okay, so the long bioassays from Lake Superior, similar results to the short bioassay in that we detected anabanopeptin A and microcystin LW, but there was no clear trends in treatment versus the toxin concentration. So at this point, all I can tell you is that we detected both of these, anabanopeptin A at 0.12 to 2.1 micrograms per liter, so higher than short bioassays. And microcystin LW in 25% of treatments is 0.06 to 0.26 micrograms per liter. And the interesting thing is that both of these were detected in almost all of the same treatments. Now, whether that means they're being made by the same species or not, we don't know, but certainly many species do make both anabanopeptin A and microcystins. So I think I got through that pretty quick, but we're right at where we want to be, I think, in terms of time. And so I think we can open it up to questions.
All right.
Yeah, great.
Let me see. So first, and I guess what I can do is I was writing down questions as the presenters were going. So I'll just start with whoever was asked when their question was asked during someone's presentation. That's who I'll go to first, and then feel free to add too. So during Dr. Sterner's, your presentation, there was just a question about just double checking something with, is this correct? With an increase of 3 degrees Celsius, the algal biomass doubled in 9 days?
That's almost correct. The 9
Okay.
days is the difference in doubling time. So the way
Okay.
I would put it is, for a given 9-day window, you would have twice as much algae being produced under warming conditions than under ambient conditions. Hopefully that made sense that way.
Yes, thank you. All right, Dr. Bullerjan, this question came while you were presenting. So we'll start with you first. Can you do these experiments with phosphorus and nitrogen with cyanobacteria and algae cell lines and then apply that data to lake samples?
You probably could, if you, I mean, there is a lot of genetic variability among blooms. So you're not looking at, even if you're looking at a microcystis bloom, there are many different genotypes existing within it that may respond differently. So I think if you had an idea that there was a bloom that was genetically fairly homogeneous, perhaps, but I really think that looking at these complex mixtures of organisms and how they interact, and also the phycosphere that's around it, the attending microbes, which are likely assisting in bloom formation. I think you really need to have the whole population in there to really get a handle on what's happening with bloom formation in nature.
All right. Any additional information, Dr. Miller, Dr. Sterner, before we move on?
I put a few comments in the chat about that question. I
Yes.
agree with what George said.
All right.
I'm not
I-
looking at the chat, so sorry.
No
No,
worries.
you're fine. Okay, this question is for Dr. Miller. Do you normalize the toxins to biomass instead of per volume?
Yeah, I'm laughing a little bit about that comment only because we did do that. So we have total cyanobacterial biomass in terms of phycocyanin concentration. And I was talking to George and Bob about this, and we've been sort of discussing how you interpret that, because obviously not every cyanobacterium that makes phycocyanin in a sample is making the toxin. It would be great if we could measure toxin genes and then divide by that maybe. But that is part of our plan, is to normalize the toxin data to something. And we have several measures of biomass that we could use. We're just kind of thinking about what would be the best variable to normalize to at this point.
All right. Let me see. Sorry, I'm like trying to double check to see. And I think I will forward the comments that Dr. Sterner had made on some of these questions. I will forward to everyone.
Yeah, I
'Cause I
didn't
think there's a
realize
few
they
peop-
weren't going out to the world.
I think it was just going to the panelists and not to everyone. So I will get those sent to everyone. Let me see. Another question that we had was, and this, I think this would be Dr. Bolerjean, but I might be wrong. I guess it is actually. Any discussion about dramatic change in cyanobium?
Cyanobium, yeah.
Yes, thank you. Between date 14 and 28.
Yeah, obviously it's there. I haven't, I really want to look at the 2023 data at the same time to see how reproducible that is, but certainly it is a remarkable change. What we do see though in Sandusky Bay, we've seen in recent years a larger population of cyanobium, and I think cyanobium is actually fairly overlooked because they're all small cells, and so if you're doing anything by microscopy, you're missing them. And so 16S is really actually revealing a large population that's always there, no matter where you look and where you go. It is striking the difference in the population between those two dates, and it's a progress report. I'll know more in a year, probably.
All right. Let me see. and I, sorry, this I think might be for Dr. Bullerjohn again. For the Lake Erie PCA that showed a separation in nitrogen fixing versus non-fixing taxa in the response to nitrogen and phosphorus treatments. Do you have ambient metal conditions at the start of the experiment, or did you modify the metals?
We did not modify the metals. That's a really good question. And we talked about doing this metal clean to begin with, and that would have been just a nightmare because acid washed everything and metal clean. That's probably stuff that should be done ultimately, but really controlling for metals in an experiment of this scope and size, just and with the people we have, we just couldn't manage. But we're not adding metals.
All right. And this, I'm not sure who it would go to, so I will just throw it out to all of you. About what about other bacterial community that exists? What other bacterial community that exists with these blooms? Do they change with the type of blooms?
Yeah, and we certainly have the data to analyze for that. All the 16S data are there, and that's what we'll be looking at. We haven't done that yet, but there are attending microbes which you can that you can align with particular species. We see attending microbes in Grand Lake St. Mary's and a Planktothrix bloom. They're different in a Planktothrix bloom in Lake Erie, and that are attached to the filaments. And so I would expect to see differences in the attending microbes in addition to the bloom. We just haven't analyzed those 16S data.
All right. Another question to any of you. How do source and non-source contributions compare between lakes, primarily focusing on land use and LODIC inputs with this question?
Well, Bob can talk about loadings from Lake Superior and what those are, those river mouths, but the dominant loading is from the agricultural landscape in Erie, and I would imagine that's the case in Green Bay as well. But
Yeah, I think Todd has indicated that CFOs are pretty important in the Fox River.
Yeah, definitely.
and they're becoming more prevalent in Erie as well. So the initial problem in Erie was row crops, but animal feeding operations are expanding in the region.
And it's a totally different universe in Lake Superior.
Yeah, you guys have trees, don't you?
There's basically zero intensive agriculture in this watershed. It's a small urban environment. We have some new data I'm pretty excited about. That there's an interesting story developing about dissolved pea. But that was one of the reasons we wanted to do this study, was that the environments are so different, yet they're all producing Great Lakes algae blooms of different kinds.
All right. Another question we got was regarding the toxin bioassays. Were the experiments only long-term, and how quickly would cyanobacteriotoxin production respond to nutrient additions? Within a few hours, one day, etc.
We were not just looking at long-term. The short-term bioassays were over the course of 48 hours. So our data shows that they do change significantly within 48 hours. Less time than that, I can't tell you right now.
Okay. So this question is for Dr. Bullerjohn. Why sample in shallow Sandusky Bay rather than out in the open Western Basin?
Yeah, I figured somebody would ask me that. The reason is that is our primary study site for the region. That's what we're tasked to do for Ohio EPA and ODNR. Yeah, we've seen a drastic change in the community over the past few years, but that's where we are. That's probably an unsatisfying answer, but that's what it is.
All right. Let me see. There was a question about how was sunlight reproduced through the tests? Did you try to reach a level to simulate on lake levels of light?
Yes, we looked at our Secchi depth and also our radiometer data to come up with what were appropriate light conditions for individual lakes. That was part of our standard operating procedure. I guess I got to talk to Bob. Bob had to leave for a seminar in another building, but if people are, I got to talk to Bob about this because he's the guy who compiled it all. But perhaps sharing the standard operating procedures with people if they're interested would be good. I don't know whether it's too soon for, if it's ready for prime time or not, but that's a possibility. I could add, the three of us could get together and say, can we share all our SOPs with everybody? It's basically like a 30-page instruction book on how to do all this.
What I can do is add, if you are all okay with this, add in the email I send to everyone with a recording to add in your email addresses so then
Yeah.
they can email you for those
Sure.
things.
Yeah.
Okay, perfect. Another question we got, and I never know how to pronounce these algae, so you're gonna just have to tell me. One question was, I understand that.
Dolichospermum. Yeah, I'm reading it.
Okay.
It's a dominant species, but does this species ratio change early versus late season that could change the differences in toxin production? Yeah, that's in Superior. So we're talking about Superior here as a dominant. So I don't, Todd's gonna have to answer that about the toxin production from the Dolichospermum we're seeing.
Yeah, so we don't actually know what toxin Dolichospermum is making. We've detected anavanopeptin A and microcystin LW in the bioassays. We've detected anavanopeptin A in the bloom samples. We don't know for sure, if Dolichospermum is making any of those. I mean, I think there's likely going to be seasonality, but also understand that the blooms that happen in Lake Superior are very ephemeral and they don't generally occur multiple times. They occur once, and so far. So any seasonality to blooms is not really known because they don't bloom more than once. So that's a really hard question to answer, I think, at this time.
All right. Another question we got was if you had insight about the impact that heterotrophic bacteria in bloom formation, toxicity, and bloom collapse.
I earlier stated we haven't actually looked at the 16S data showing how the populations may change as the bloom forms, but there are increasing the importance of the phycosphere cannot be underestimated. I mean, you can't think about cyanobacteria acting in isolation. There is a attending microbial community which is present and is likely assisting in bloom formation and ultimately decline. Our lab studies mechanisms of bloom decline caused by fungi, perhaps, as an example, from chytrid fungi, phages, and all that. So, bottom line is, I can't give you any hard answer now, but you cannot view the community without the heterotrophic bacteria, without the phycosphere, the microbiome, whatever you want to call it.
All right. I think that was the majority of the questions. I'm just looking at this quickly. I don't see any others that we have not discussed. I was trying to get the questions the answers that Dr. Sterner had responded to into the chat for people to take a look at. I'm not sure I'm as successful with that as I had hoped to be. So I may not be able to do that. I will try to do this. Yeah, sorry, I can't seem to do it. All right, I think those were all the questions that we had. And what I will do is I will make sure that people get the recording as well as everyone's contact information, so then they can get that protocol that you had mentioned, Dr. Bullerjean. So let me wrap up since we are right at 1 o'clock. I wanted to thank again, Dr. Bullerjean, Dr. Miller, Dr. Sterner at his seminar for their willingness to talk about their Sea Grant research. This has really been an excellent discussion. I did want to remind everyone that there is a webinar survey URL in the chat feature, so please take a few minutes to fill that out. This webinar series is sponsored by Ohio Sea Grant and will continue on in May with Drs. Holler and Kennedy from the University of Toledo, who will be talking about a potential treatment for health effects of algal toxin exposure. The registration link is in the chat. Again, thank you, Dr. Bullerjean. Thank you, Dr. Miller. Thank you, Dr. Sterner, and all the participants. We really appreciate you being willing to talk to us about this great research, and we're looking forward to the part 2 to this discussion. So thank you again. This was really beneficial. We hope that everyone will be able to attend an upcoming webinar soon. Thank you again. Thanks again, Dr. Bullerjean.
Yep, thanks, my pleasure.
Thank you.
Really appreciate it. Thank you. Have a
Bye-bye.
great afternoon.
Bye.